Fast-acting vitamin D receptor agonists for the treatment of IAC conditions, infection-associated chronic conditions. Calcitriol versus paracalcitol, Part 2. In this YouTube series, we are discussing three main topics: the nomenclature and structure of fast-acting vitamin D receptor agonists, which include the endogenous hormone calcitriol, 125 vitamin D3, and its synthetic analog, paracalcitol, 125 vitamin D2. We are also discussing the medical properties and medical indications of vitamin D receptor agonists. In particular, we are discussing the possible repurposing of paracalcitol for the treatment of COVID and long COVID. The goal in these discussions is the potential augmentation of the physiology of remission, not only of long COVID, but also other IAC conditions. Long COVID may cause long-term changes in the heart and lungs and may lead to cardiac and pulmonary diseases. Mount Sinai studies suggest COVID-19 infection should be considered a risk factor for future cardiopulmonary conditions. It is now known that COVID infections deposit numerous COVID toxins such as the spike protein, envelope protein, and viral RNA in numerous tissues and organs within the body. These lead to toxic reactions, histopathological damage, as well as prolonged inflammation. Today we will discuss the role of some specialized lipid metabolites which are derived from arachidonic acid, which is frequently produced in inflammatory conditions. Arachidonic acid is very pro-inflammatory, and some of its derivatives, lipid metabolites, such as prostaglandins and leukotrienes, are also pro-inflammatory. In contrast, when a specific set of enzymes acts on arachidonic acid, a set of anti-inflammatory lipid products are generated. These lipid metabolites are known as EETs, which is an abbreviation for a complex word: epoxy-ico-citrianoic acids. EETs are generated by a set of enzymes which include an enzyme known as CYP2J2, CYP2J2. It is a member of the cytochrome P450 superfamily of enzymes. There's 57 of these P450 enzymes in the human genome. CYP2J2 is primarily localized to mitochondria. Its role in generating these anti-inflammatory lipids, antioxidant lipids, EETs, makes CYP2J2 cardioprotective. In addition, we will see new evidence that CYP2J2 is also renoprotective. I have started this animation to emphasize that during viral infections and IAC conditions, viral particles, viral toxins, as well as inflammatory mediators are pumped through the circulatory system through the cardiac chambers, where they can impact cellular functions and structure, initiating long-term damage. For that reason, it's necessary to locate, identify off-the-shelf therapeutics that can be used to attenuate these inflammatory pro-oxidative stress metabolites and counteract them. For that reason, agents that can boost EETs will be very beneficial in these actions. In addition to being cardioprotective, CYP2J2 is also renoprotective. And as we will see later in this YouTube, it is now known that paracalcitol, by activating vitamin D receptors, can upregulate the expression of CYP2J2. And we can represent that with that green information vector in the center part of this slide. 125 vitamin D2 interacting with the vitamin D receptor produces pleiotropic actions, multiple actions, one of which is the upregulation of this cytochrome P450 enzyme, CYP2J2, which serves to take lipid metabolites derived from arachidonic acid and convert them to the anti-inflammatory lipid metabolites, EETs. What are the role of ETTs in COVID and long COVID? Here are two references, one from 2022. The role of cytochrome P450 enzymes in COVID-19 pathogenesis and therapy. Cytochrome P450 genes expression in human prostate cancer. So with 57 genes in the human genome, these interactions are complex. This illustrates the conundrum that scientists and doctors are facing, how to manipulate these pathways to promote healing. And that's what we're going to talk about today. And this story goes back over 20 years. In the upper part of the slide, there is a panel from this paper, Enhanced Post-Ischemic Functional Recovery in CYP2J2 Transgenic Hearts. These are transgenic mice, their hearts. Involves mitochondrial ATP-sensitive potassium channels and P42, P44 MAP kinase pathway. To better understand the significance of the information vector highlighted in green, I would like to do a deep dive into a couple of the figures of this paper from 2004. First, on the left, you see some fluorescent images. These are taken with a laser confocal microscope. You're looking at the fluorescent properties of cells, cardiomyocytes, that have been derived from the hearts of these transgenic mice. So just for nomenclature, if we read the legend for figure 5, endogenous flavoprotein fluorescence in cardiomyocytes. These are the cells from the heart. Cardio, heart, myocytes are muscle cells. From CYP2J2-TR, which stands for transgenic mice. So these mice have been genetically engineered to overexpress CYP2J2 in the heart, specifically heart muscle. Compared with wild type, WT stands for wild type mice. A, representative confocal images. These are laser confocal microscopy, a form of fluorescence microscopy, showing flavoprotein fluorescence. This is endogenous fluorescence. No dyes have been used. There's no green fluorescent protein in these transgenic animals. We're looking at the flavoprotein fluorescence in wild type and the transgenic cardiomyocytes at baseline before they've been stimulated. And what are they going to be stimulated with? EETs. And so before stimulation, if you compare the wild types, which have moderate endogenous flavoprotein fluorescence, compare them to the CYP2J2-TR transgenic mice, these cardiomyocytes have much more flavoprotein fluorescence. This is because this protein is being overexpressed, and once again, this protein is primarily localized to mitochondria, which are densely present in cardiomyocytes. To the right, you see the molecular structure of flavoprotein. So when you irradiate it with, say, green or blue light, it will fluoresce. And that's the light that's being imaged in these confocal micrographs. Now, further to the right, you see the structure of an EET. And this again stands for an epoxyicosahexaenoic acid. A sidebar comment. Note that at the left portion of this image of the EET, you see an oxygen molecule represented with an O, and its two chemical bonds are linked to adjacent carbon atoms within the lipid. And this type of configuration, where an oxygen bridges between two carbon molecules, is known as an epoxy. This configuration, the cell has utilized an enzyme to create this epoxy bond for a purpose, and that is to change the signaling properties of these lipid molecules away from being pro-inflammatory to be the opposite, anti-inflammatory. So that epoxy bond is critical. Now above that, you will see an abbreviation for two EETs, which were used to stimulate the physiology of the cardiomyocytes from these transgenic mice. So the term 14,15 indicates the carbon position of the epoxy bond. So where that oxygen, which has bonds to the adjacent carbon atoms, that's the 14 and 15 carbon atoms of the EET. That's one type of molecule that was used to stimulate the cardiomyocytes. In contrast, another EET, 11,12 EET, and that's where the epoxy bond bridges the 11 and 12 carbon positions of that molecule. It too was tested to compare and contrast with the actions of 14,15 EET. Now, the graphical summary of these experiments shows first the control fluorescence. These are the cells derived from the heart of the transgenic mice, which have been overexpressing CYP2J2 in their cytoplasm, and once again, this protein localizes primarily to mitochondria. So when you look with a fluorescent microscope, looking at the endogenous fluorescence of the cells from these transgenic mice, they have a certain basal fluorescence. Now, take those same cells and stimulate them with these two different types of EETs. First, 1 micromolar, and there is a dramatic boost in the fluorescence from the cells. That indicates that there's been a further upregulation of CYP2J2 in response to the stimulus, the EET, in this case 14,15 EET. To the right of that, if you add more, 5 micromolar of this 14,15 EET, you get an even larger boost in the expression of flavoprotein fluorescence. This indicates a more robust response in this critical protein, which serves as an antioxidant agent for the cardiomyocytes to withstand oxidative stress. Now, if we move to the furthest to the right, we see the effect of 1 micromolar of 11,12 EET. So this epoxy-eicosapentaenoic acid with the epoxy group at the 11 and 12 carbon positions is a stronger agonist to stimulate the upregulation of this flavoprotein within these cardiomyocytes. And once again, this flavoprotein plays a role in the antioxidant capacity of the cells. And this is one of the ways that EETs change cell physiology from pro-inflammatory responses of cells to anti-inflammatory responses of cells. So from Wikipedia, here's the description of flavoproteins. Flavoproteins are proteins that contain a nucleic acid derivative of riboflavin. These proteins are involved in a wide array of biological processes, including removal of chemical radicals, contributing to oxidative stress, photosynthesis, and DNA repair. The flavoproteins are some of the most studied family of enzymes. Here is an AI overview of EETs. Epoxy-icosatrienoic acids, EETs, are signaling lipid mediators synthesized from arachidonic acid by cytochrome P450 epoxygenases. They are potent vasodilators and anti-inflammatory compounds that promote cardiovascular health, angiogenesis, and cell survival, primarily metabolized into less active dihydroxyicosatrienoic acids, DHETs, by soluble epoxy hydrolase. So that's an enzyme that inactivates them. How do EETs pertain to COVID and long COVID? This is a very important paper from 2022. Serum levels of pro-inflammatory lipid mediators and specialized pro-resolving molecules, those are molecules that prompt remission of inflammation. So specialized pro-resolving molecules are increased in patients with severe acute respiratory syndrome, coronavirus 2s, SARS-CoV-2, and correlate with markers of the adaptive immune response. So these pro-resolving molecules are EETs. At the bottom part of the slide is an AI overview of the properties of EETs. They have anti-inflammatory and protective effects. EETs demonstrate significant anti-inflammatory actions by inhibiting leukocyte recruitment and signaling pathways. They are cardioprotective, reducing ischemic damage and aiding in tissue repair. Very important for long COVID and other IAC conditions. Prompting tissue repair. This is boosting the physiology of remission. Therapeutic potential. Inhibition of the SEH enzyme. This is the enzyme that inactivates EETs. So by inhibiting this enzyme, the EETs remain around longer, boosting their anti-inflammatory, antioxidant signaling pathways. So inhibition of the SEH enzyme, which breaks down EETs, is a therapeutic strategy being studied to increase EET levels for treating hypertension, diabetes, and heart failure. Sidebar comment. These inhibitors of the SEH enzyme might in the future be useful for the treatment of long COVID as well as other IAC conditions. Role in disease. While it is generally beneficial in cardiac health, elevated EETs have been linked to promoting tumor angiogenesis and metastasis in certain cancer models. These are animal models. And it's important to comment here that any of the therapeutics that are being used for COVID and long COVID are going to have side effects and counterindications. And so generally, physicians are concerned about vitamin D receptor agonists for having unseen actions. For instance, promoting cardiovascular health, that's a benefit, but if it's promoting tumor blood vessel formation, angiogenesis, or promoting metastasis, that's very serious counterindication. However, the counterpoint to that counterpoint is that paracalcitol has been used for 30 years in chronic kidney disease patients. And I have yet to read about a cancer signal associated with the use of paracalcitol in this cohort of human patients. And so as scientists discuss these issues with physicians, we have to zero in on these potential unwanted side effects, but at the same time recognize that the study of the use of paracalcitol in chronic kidney disease patients as they acquire COVID and long COVID, and they continue to be treated with paracalcitol, that doing retrospective studies should be able to answer whether paracalcitol poses any risk. So with that, I'd like to move to this key new paper, which reports on the benefit that I was speaking about earlier in this YouTube, how boosting CYP2J2 is renoprotective, protecting the kidneys. Now here are 3 information vectors. We've used the top information vector in a number of prior YouTubes on this channel. Using paracalcitol or calcitriol to stimulate vitamin D receptors, I'm just going to refer to them both as 1,25-dihydroxy vitamin D, 125D, stimulating vitamin D receptors to upregulate the master anti-inflammatory transcription factor, NRF2, which upregulates many genes, including GPX4, glutathione peroxidase 4, a key enzyme involved in the suppression of reactive oxygen species and peroxidated lipids, the upstream drivers of programmed cell death known as ferroptosis. So suppressing ferroptosis is a key physiological benefit of these vitamin D receptor agonists. And we see that they're being repurposed to accomplish this, to suppress oxidative stress and ferroptosis in a number of organ systems. Below that is a new information vector which we're introducing in this particular YouTube series, concentrating on CYP2J2, the cytochrome P450 enzyme, which creates these important anti-inflammatory, antioxidant signaling pathway lipids, EETs. So stimulating vitamin D receptors with paracalcitol, identifying that as 125 vitamin D2, that's the abbreviation for paracalcitol, binding to a vitamin D receptor, upregulating CYP2J2. CYP2J2 enzymatically converts arachidonic acid into 4 types of EETs. We will mention that detail shortly. These EETs interact with receptors. In the case of macrophages, it changes their behavior from pro-inflammatory to anti-inflammatory. More importantly, these second types of cell behaviors are known as M2 polarization, and the macrophages that express this phenotype are called M2 macrophages, and they promote tissue healing, which is the key goal, which we mentioned at the beginning of this YouTube, the use of vitamin D receptor agonists, particularly paracalcitol, to augment the physiology of remission, which includes tissue repair. So the information vector of EETs stimulating the shift of macrophages to their M2 polarized phenotype, and polarization refers to their epigenetic trajectory. So once you have these M2 macrophages in a tissue, they help suppress inflammation. And in this case, they suppress progressive diabetic nephropathy, kidney injury. So using this as background information, we can now read the title of this paper and it will make more sense. Paracalcitol ameliorates, which means makes better. Paracalcitol ameliorates diabetic nephropathy by promoting EETs and M2 macrophage polarization phenotypes, and inhibiting inflammation by regulating vitamin D receptor CYP2J2 axis. And this term axis is a phrase or a semantic that comes out of endocrinology and immunology. It's a way of saying molecular agents or molecular agents interacting with genes, the set of interactions, in this case, stimulation of a vitamin D receptor, an active vitamin D receptor, can upregulate the CYP2J2 gene, leading to the upregulation of the protein, an enzyme. So that's the VDR-CYP2J2 axis. The net result is that CYP2J2 will act on arachidonic acid, a pro-inflammatory lipid produced during inflammation, convert it to EETs, which are anti-inflammatory lipids, and also triggering antioxidant responses such as the upregulation of flavoprotein, a key antioxidant molecule in cells. So CYP2J2 is a primary human epoxygenase, an enzyme that puts epoxy groups on molecules, in this case arachidonic acid, highly expressed in cardiovascular tissues, that metabolizes arachidonic acid, AA, into 4 regioisometric epoxy-icosatrienoic acid, EETs. Now, regioisomeric means the enzyme or the variety of molecules that can result from an enzymatic reaction, in this case where the epoxy group is placed on the arachidonic acid, can vary, and 4 major metabolites are produced from arachidonic acid through the actions of CYP2J2 enzyme. And these 4 types of metabolites, these lipid metabolites, these EETs act as signaling molecules to promote cardiovascular health, including vasodilation, anti-inflammation, and cardioprotection. We should also add now renoprotection, at least against diabetic nephropathy. CYP2J2-mediated arachidonic acid metabolism is a key target in cardiovascular diseases and cancer. And again, it's important to emphasize that the role of these EETs in cancer needs to be addressed in chronic kidney disease patients to better understand how pericalcitol could potentially act on long COVID patients as well as patients with other IAC conditions. Cytochrome P450 2J2, abbreviated CYP2J2, is a protein in humans. It's encoded by this gene, and the nomenclature here follows convention. A gene is italicized, whereas the protein is non-italicized. So the protein CYP2J2 is a member of the cytochrome P450 superfamily of enzymes. The enzymes are oxygenases, which catalyze many reactions involved in the metabolisms of drugs and other xenobiotics. Those are chemicals that are foreign to the body. CYP2J2 is also involved in the synthesis of cholesterol, steroids, and other lipids. So this inset figure illustrates the 2 classes of macrophages that immunologists just discussed. It's a simplified construction of pro-inflammatory macrophages versus anti-inflammatory macrophages. M1 phenotypes are the pro-inflammatory macrophages. M2 phenotypes are called anti-inflammatory macrophages. So the polarization of behavior refers to whether stimuli shift the macrophage to expressing this M1 phenotype, or alternatively, other stimuli such as EETs converting the phenotype from M1 to M2 phenotypes, and that promotes tissue healing. And there are molecular markers that immunologists use to define these phenotypes. So M2 macrophages, or, quote, alternatively activated macrophages, are specialized immune cells that drive tissue repair, remodeling, and anti-inflammatory responses, largely activated by interleukin 4 and interleukin 13. They contrast with M1 macrophages by promoting healing and tumor progression rather than fighting microbes. M2 macrophages are generally identified by surface markers such as CD163 and CD206, two surface markers. So in part 3 of this discussion, we will go into the details of this paper we just mentioned, how paracalcitol can ameliorate diabetic nephropathy. If you would like a PDF slide summary of this particular YouTube, which contains active links to other YouTubes on this channel, please go to the Google Docs folder highlighted in green at the bottom of the slide. This link will take you to a set of folders. If you'll navigate to the subfolder called Download First, you'll be able to locate this particular YouTube's PDF slide summary more quickly. Also, in this Google Docs folder, you will find subfolders which contain key references to topics like COVID and ferroptosis. Earlier this year, we posted a number of YouTubes on this channel on the topic of ferroptosis being driven by GPX4 gene silencing, which occurs when the promoter region for the GPX4 gene becomes hypermethylated. And this can occur in response to ischemic reperfusion injury in the liver, and it may also be occurring elsewhere in the body. This is particularly important for COVID and long COVID because of the prevalence of microclots, especially in long COVID patients. When these microclots are dissolved or dislodged, that tissue is likely to have ischemic reperfusion injury. And this type of injury creates reactive oxygen species, which can in turn peroxidate lipids and lead to ferroptosis, programmed cell death. So these prior YouTubes discuss the potential utility of utilizing vitamin D receptor agonists, particularly paracalcitol, as a potential way to reduce ischemic reperfusion injuries in long COVID impacted tissues.